let's get started. So this section is going to be about ECG, which is like one of the most common medical signals we have, I guess, I don't know. But we will be focusing mainly on a signal classification problem. And I also prepared some slides for you, but we're gonna spend like about 15 minutes on slides and then turn to demo later. So let's get started on ECG. I'm from MeDA, so yeah, I'm Zhongqi, and signals are really common, and we do have like a bunch of signals in real life. And so here is some introduction on how to proceed with signals. So a signal is basically a function that contains some kind of information about something. And so you can kind of regard everything as a function pretty much. So pretty much everything is a signal. So for example, we have audio signals, we have image signals, which usually we wouldn't call an image a signal because it's like kind of weird. But an image can be thought of as a function in which that each pixel was assigned a value to it. For example, like some kind of intensity on a color or something. So an image can be considered a signal. And usually there are like two types of signals. The first one is continuous time signals, which are pretty much all the in real life signals. And we also have discrete time signals, which we will be using because like if you're doing calculations and stuff, you couldn't really store like an infinite amount of data in memory. So we're going to use discrete time signals here in the demo later. And so the main topic here is the ECG signal, which records heartbeat for you. So I've attached a random internet signal there, so for your reference. Okay, so what is ECG? I am not sure whether you have learned what an ECG is or like what, how do you call an ECG, each segment of an ECG signal or something. But so I'm going to talk a little bit about ECG first before we head into the Colab tutorial. So an ECG signal is basically like a recording of heartbeats. And so you can kind of decompose an ECG signal. For example, like if it's one heartbeat, then it's going to be in the form of the picture on the right. And you can kind of decompose it into different components. For example, like P wave, like a little hill in front of the signal, and then we have like some sharp turns, which are the QRS complex. And you can see there's a huge R above the one crazy peak there. So that's called the R peak. And we also have a T wave, just like the last wave over after the QRS complex. So they all depict different process of a heart beating, but it's not really important in today's lecture. So yeah, so we're gonna just focusing on the nouns and how we call them. And so ECG can be recorded in different ways. For example, like we have, because when you record the ECG signal, it's like you place a lot of different electrodes on your body, and it means that you can kind of, detect an ECG signal in different points of view. So these are called leads. So we have 12 leads usually for one ECG signal, and it's a little bit complicated, but we are not really going to be using the exact definition here. So this is just for introduction. So what are the 12 leads? So we have lead 1, lead 2, and lead 3. So on the picture, it's going to be on the leftmost of the picture. And we also have AVR, AVL, right and left, and inferior AVF. And we also have chest leads, which are V1 to V6, which is the rightmost 6 ECG signal in the picture. Yeah, so these are the 12 leads of ECG. And so the main topic or the main problem we're going to solve today is a detection on atrial fibrillation, which can be abbreviated to AF, and we're going to call it AF later. So what is atrial fibrillation? So atrial fibrillation is a heart disease. I think it's a heart disorder that is can be characterized by usually irregular or rapid heartbeats. For example, so you can see on the ECG pattern here that it consists of irregular RR intervals. For example, like, so in the following images, the right one is the normal ECG. So in the bottom right signal, you can kind of see that the RR interval, which is the distance between two consecutive R peaks, are pretty much exactly the same, right? It's about, they're about 0.8 seconds, which is pretty regular. And on the left, you can see that if it's AF, the ECG signals is going to be a bit distorted, and the intervals are not really exactly the same. Like the here, you can kind of see it's shorter, and you can kind of see it's like a little longer here, so it varies. So these are one of the characteristics of AF. And the other characteristics of AF is the absence of distinct P waves. So what are P waves? So P waves are just what I just said. It's a little hill in front of the QRS complex. So you can see that here in the normal ECG signal, the P wave is here present, and it's pretty visible. But if you look at the AF ones, then you can kind of see that the P waves are not really here. Like it's barely visible. Here it is. This is probably P wave, but I'm not sure. So one of the characteristics of AF is that the P waves might be disappearing. Like it might not be existent. So yeah, so these two are how humans or I usually see, like, if we want to verify if a ECG signal is AF or not. And so we're not gonna use this today, but this is just for reference. So these are called heart rate variability or HRV features. So what is heart rate variability? So what we just said, that if it's an AF signal, then the heart rate is going to be irregular, right? So if we somehow measure the variation or the volatility of the heart rates, then it can be somewhat deduced that maybe we can kind of try to classify each signal this way to just calculate something. So these are called HRV features. So it tries to record all the RR intervals and try to calculate some kind of variability out of it. And there are a lot of different ways to calculate volatility or variation. So there are a lot of nouns here, but this is just for introduction. So I have like a Wikipedia link. So if you want to Google or something, you can just check that out. So different types of HRV features can include like time domain features, which is just what I just said. For example, like SDNN is the standard deviation of NN intervals, and NN intervals are just normal RR intervals. So this is just some kind of standard deviation. And we also have frequency domain features, which are obtained after applying some kind of mathy function to the signals. So then you kind of obtain a pseudo distribution-wise thing on the signal. And so we have low frequency ones and high frequency ones, and also the nonlinear ones. So these are probably some kind of things that you want to be knowing or checking out if you are interested in the problem of the classification of AF. Okay. Yeah, and so here is some special traits of ECG signals in machine learning, just before we head into the actual demo. So for example, like the ECG signal is really an interesting topic because it's one-dimensional, which is which means that it can have a lot of advantages if it's like compared to like images or MRI scans or I don't know, CT scans. So it uses less time when you load data or when you process them, and it is lighter, so you do not really need to do resizing or resampling because it doesn't really eat up that much memory in your computer. So it has some kind of easier adaptation to some like front-end models, not front-end, cutting-edge models like for example, like language models maybe, because like you can kind of regard like a sentence as a some kind of signal, I guess. So because it's one-dimensional, so it has some advantages here. And also being a signal means that you can kind of view it in different ways. For example, like you can view it as a one-dimensional image, which means that if you have like a some kind of transforms or something you can do with an image, you can deal with signals as well. For example, like if you want to apply some like Instagram filters on your images, you can do it on signals, or you can do some contrast, I don't know, contrast resampling or something on signals too. So that is one of the advantages. And also you can view it as a signal. So there's a specific field for that, for processing signals. It's called signal processing. And it has some interesting mathy things in it. And so, for example, like bandpass filter or continuous waveless transform. And also you can view the signal as a time series, which means that you can apply some statistical things to it, like AR, like Array-Max model, moving average model, or like calculate the autocorrelation, like try to like use the periodicity of the ECG signals to your advantage. So yeah, so that's pretty much the intro to ECG, and we're gonna head into the demo here. So the demo is, which is a classification problem, is it's gonna the objective is to classify 30-second ECG segments into two categories, AF or non-AF. So we also have other cardiac conditions here, but they are merged into non-AF. So non-AF means like normal and possibly other conditions. And yeah, so how are we going to do it? We're going to use machine learning, more specifically deep learning, with ResNet 18 as our model. And so we're going to train the model on Google Colab, and for your amusement, I guess. And the workflow is going to be load the data, pre-processing, and do training and validation, and do testing on a separate test set. Okay. And the data we're going to use is an open data set from, it's called MIT BIH AF database. Yeah, it's a pretty well-known database on ECG AF detection, and it has two lead, so it's like a two-channel thing, 10-hour ECG signal from 23 patients, and each ECG signal is sampled at 250 samples per second. That means that because our goal is to try to classify if a 30-second segments are AF or not. So a 30-second signal means that it will be containing like 7,500 values in it, because like 250 values per second. yeah, it has both normal ones, AF and other recordings. And I've already cropped it into 30-second segments, so because the code is kind of messy. So I'm not going to include that in the demo. And these are the links. You can kind of try to look at the data set we're using. Okay, so as for the preprocessing, there's something like these three things that I want to talk about. So the first two is pretty standard way of dealing with ECG data. The first one is removal of Gaussian noise. Gaussian noise is just like a white noise. So it happens during measurement. So we're going to remove that. And usually we want to remove baseline wonder, which is as the picture on the right shows. So with baseline wonder, you can kind of see the ECG is roaming into the space or something. And then without baseline wonder, it's like a straight line. So if we feed the model with something more regularized, that might be better for the model to learn the important features instead of the random roaming. Yeah. And then the continuous wavelet transform is something I talked about before. It's a mathy function that takes in a ECG signal and transform into some kind of distribution on the frequency domain, which means that it's a distribution of frequencies for each time steps. That doesn't really mean anything. If you don't really know anything about continuous wavelet transform, you can Google it if you want to. Okay, so the model we're using is ResNet 18, which is a bunch of matrix multiplication, basically. So we have, so the leftmost, we have input. So we input a signal, a two-channel signal into the model, and then do a bunch of matrix multiplication and do some stuff, and then output a possibility on whether it's AF or not. Okay, so it's demo time. So we are gonna head into the IPython notebook here. So please open the IPython notebook. And so after you're in this page, So after you're in this page, you kind of want to save a copy in Drive, because so you can edit it. So after you've saved a copy, you have to because we're doing training, so you have to change your runtime type to GPU. So pick this option here, T for GPU, and then click save. Okay, so now we're ready. So the upper part is just what I just talked about before. So first thing first, we are gonna load our data. So how do you, because some limitations of Google Drive. So you have to add a shortcut here by clicking right clicking and pick add shortcut. And then you want to choose, for example, just my drive. Just my drive. Add it into your drive. And click add. And then it should be in your drive, in my drive, I mean the folder. And then, so this is going to be the same from the previous lectures. Click the little run button here to mount your drive, to connect your drive into the notebook. So click your account and click continue. Takes 10 seconds or something. Okay, so now I guess you've already mounted, so you can click the folder icon here to view the stuff in your drive. So you can kind of see that the the shortcut we just added here is right here. So mitbih.af.zip. And then after you've mounted your drive, you want to unzip it, right? So click this run button, and then it'll say that it's inflating or something, and then it's done. Okay, so you've successfully inflated, unzipped it, the data into your current work environment, which is not in your drive, which is in like some cloud server in Google. So here you can see in the folder that we just unzipped, we have, like, I've left one raw signal just for reference, and I have, because I've already segmented the signals. So here in the process data, we have a bunch of 30-second ECG signals. Okay, so first, let's install the packages we're going to use. So MeDA, yeah, MeDA is like for medical imaging, it's pretty useful. And we have WFDB, which is for this data set, because this is from PhysioNet. And the data from PhysioNet usually use this library to help visualize or read or write, I guess. So install it. After you installed it, you have to import it, obviously. Yeah, and then because the main reason why I left a raw signal inside the folder, because we are trying to look at the raw signal first. So we are gonna load this data 40043, and then try to look at, what it's like. So here, the signal means that the ECG data. The annotation means the corresponding label. For example, the whether it's AF or whether it's like normal or other things is in the annotation object. So timestamps here means the timestamp at which the labels happened. And the labels are of the same length as the time stamps. So you can see the length is 166, which is the same as the length of the timestamps, which is 166. So each label here corresponds to one timestamps. And it means that starting from the 68 value, the ECG signal is normal. So after like a while, after like this number, the ECG signal is going to be AF instead of normal. So that is how you look at the annotation file. So because the raw signal is of 10 hour, so the signal length is going to be super long, which is here, the number here. That is normal. So first, we're going to plot it by clicking this. So the colors means that, it's better to look at. And then you can kind of see there's a cramped, messy diagram here, graph. Because it's way too long, it's 10 hours, so it doesn't really do anything. Like if you plot it, it doesn't really tell you anything. So you kind of want to zoom in. For example, here we only plot like from the half of it to the 50% of it, to try to look at what it looks like. It's still really messy, so we're trying to going further. So here, yeah, this is like a nice example of an ECG segment. So you can kind of see there's a red line here. That means that after this red line is going to be AF, and then before this is going to be it should be normal or other conditions. So like what I just said in the slide, you can kind of see that when it's AF, there's some really irregular stuff happening here. Like the QRS complex is really weird, and the P waves are not really existent anymore. So yeah, so this is how you visualize or read the raw signal from the database. And then we are gonna jump into the training section of it, which is to use the process data from the zip file I just gave you. The data path is this, and then you try to get the file names right, and then you can print it. So these are the IDs of the raw file names. You can kind of think of each ID as one patient. So this guy, this person, 4015 is a patient, and then 4043 is a patient. So we have 21 patients because two of that is not available. Sorry, we have 23, I think, yeah, 23. And then we are gonna, because we're gonna use some shuffling when we head into training. So we want to make sure the random thing that's gonna happen, that we're gonna make sure that it is fixed instead of re-random. So just set determinism so you can try to reproduce what we're doing here. You can also not do this. Like if you don't run it, then it's gonna be different each time you run this notebook. Okay, so now we've fixed the randomness. We are ready to split the file names into training, validation, test sets. Please note that we have not yet load the files yet. The reason why we do split here is because you do want like test set to be of like a different patient from like the training and validation sets. You don't want like some samples of a patient to be in training but also in testing. That doesn't really make sense, since the testing should be really independent of the training and the validation. So here we're using the IDs of the file names as like some kind of identifier of the patients so that we don't get messed up. The training portion is going to be 80% and the validation portion is going to be 10%. That means that we have 10% left for test. And so yeah, so here's some syntaxes, just run it. So we have 18 files for training, 2 files for validation, 3 for testing. And then there are some caveats to it. Since 719, this file and this file is not like they don't really contain anything other than AF. So like if you use it for validation or test, it's gonna mess up the output pretty much. So because like the validation only consists of 2 files, right? So if you use this for validation, it's gonna be like 75% AF or something. The imbalance would be immense. So we are gonna use these for training only. That means that we're gonna remove them from validation or test and swap something else in. So after you run that, you can load in the samples. So here the first 5 samples are these, and we have a total of 4,400 samples. That means that we've sampled 200 samples out of 200 samples out of 1 patient. Yeah, so it's now ready to be loaded. So if you look at the files here, file names here, you can kind of see that this is the patient ID or the identifier, and this is the label. Like 1 means that it's AF, 0 means it's not AF, and 1,000 is this number here means the number of files, the index of files generated, right? Because we have a total of 4,400 data. So this number means that it's the 2,249 sample to be generated. That was generated. Okay, so like we usually do, let's plot the first sample to see what it's like. It's always a good idea to see what the sample is like. So you can kind of see that, you know, it's much shorter since it's like only a 30-second signal. And we also have two numbers here. The start means that this corresponds to the placement, the original timestamp in the raw ECG signal file. So this means that it's starting from here to here. So it's like this. And because we're using two lead ECG signals, so this is the first lead, or channel 0, the first channel. Lead are usually called channel in machine learning. This is the second channel. So they are different because there are different points of view of a same ECG signal. And yeah. And now we are ready to load the data into different sets, just like we did before. So this is messy, but it's fine. So after we've split the data, we can now see that a total of 4,400 samples have been put into different sets. Like we have 3,400 in training, and we have 400 in validation, 600 left for testing. And to kind of give you an idea on what this stuff contains, this is basically a list. These are lists. And it has a bunch of dictionaries that has references to the original signal. Please note that we have not loaded the signal yet. We only represent it using the path here, so you can kind of see that it's a path, it's a string, it's not really some kind of number yet. So maybe we just look at the last item in the dictionary. You can see the signal is this, and the label is 0, because this is 0, yeah. And this is exactly what you want when you do machine learning or deep learning, because you don't really want to load the data at all, like at once. I mean, because the memory is not going to be enough. And just for training purposes, we are going to initialize all the models and like optimizers or something using PyTorch, which is a really common library for machine learning or deep learning. So import it, and then we are going to use GPU. So that's why we changed the runtime into GPU, and so it's faster. And we're going to use ResNet 18, which has a dimension of 1, since ResNet is capable of doing data regardless of its dimension. So here ECG signals are of 1 dimension. And we have 2 leads. So the input channel is going to be 2. And we are trying to predict if it's AF or not, right? So it's 2 classes. And then we have optimizers. So optimizers are just something that helps you update your model. So you couldn't really update the model by yourself, correct? Right? Because it contains a lot of parameters. And we have a loss function, which is basically we're trying to tell the machine to see if the prediction is close enough or not. So we have a loss function, and we're trying to minimize it. So we're trying to make the machine work better in terms of prediction. And we can see the output here is basically I printed out all the stuff in the model. So you can kind of see the structure of ResNet 18. It has a convolution layer, which is some matrix multiplication and some matrix multiplication, and then some function, and then a lot of blocks. And then after all these, it'll output 2 numbers, pretty much the class probability of the two classes, which is AF or not. So that are the models and the technical stuff. And now we're ready to pre-process the ECG data. So here I've prepared three basic, I don't know, standard practice for you, which is the noise removal I just talked about in the slides, and the baseline wonder and align center. So noise removal removes white noise. Baseline wonder removes baseline wonder, and then align center means that it centers the signals, so it does not really go into like randomly higher levels or random high levels or like or be too away from the center. That might be a little concerning for the models. So these are the implementation of it. You just press run to register it, and then the following set of transformations are crucial for loading the image, obviously, because we have not yet loaded the image. And then we might want to resize, but maybe you can skip this. I don't know, because signals are not really using that much memory. So if you don't resize, that's pretty much okay. And then maybe you want to add some like random operations to it. For example, like random spatial crop. That means that this function randomly crops the loaded image or loaded ECG signals for you. So add more noise basically. And then random Gaussian noise pretty much does what it sounds like. And to tensors, D means it's trying to put it into tensor, which is the data type that's going to be used by PyTorch. So here we're going to import it and import the transforms we are going to use. And this is the official transforms that we are using today. So you can kind of see it loads the image. Sorry, it loads the image means signal. Loads the signal and then it resizes it. So because the signal is a 30 seconds, so it contains 7,500 values. So maybe we want to reduce that into just something like this, 2048. Then it's kind of like shorter, so it's maybe lighter and it'll compute faster. And so do some noise removal, do some baseline wonder removal, do some align center, and then maybe you want to do some random stuff, maybe you don't. So I've commented out the operations here, but you can uncomment. Just remove the hashtag in front of it to uncomment it if you want that to be alive. If you don't want that to be alive, then you can just comment it off. Maybe we should try to look at the output of some standard practice, see what it's like first. So I will uncomment that later. Okay, so now it's like I've run it, and so let's look at the effects of the transformations. So So this is the first training sample. This is the plot, and then we have a. So you can see the effects of the transformations. So, for example, that like this is kind of noisy, right? It's like really messy inside, but this is like much better. The noise has been removed. A lot of noise has been removed, so it might be better to remove noise. And also you can see that the x-axis is originally 7,500, right? Just as I said, but now it's like 2,000 and more. So it's much better in terms of efficiency. So you can kind of alter the transformations to see the differences. For example, like maybe I uncomment this and press run, and then you can kind of see the effects of the transformations. You see that it's like it's cropped really short. It's cropped to like barely 1,000. Sorry, a little more than 1,000, and it's like with some really weird shapes going on because we added noise. So this is the original thing. This is the transform thing. Like even the intensity is not the same anymore. So you can try to mix and match all the transforms to get what you want. So sorry. So let's get rid of the random first, because like we want to see the baseline performance of the model. So this is the transforms without randomness. And then, so here's some techie stuff again. So we have data sets, which helps us load the data. We have data loader that helps us load the data. And then, so it takes some time, but it eventually loads. And we have like data sets here. So the commented section means that when you do not have enough memory, you might want to not use the cache data set, because cache means that it stores like a temporary copy in your memory, and that might be really expensive sometimes. I don't know. So maybe you sometimes want to use data set instead, or you want to lower the cache rate to like half, maybe. But Google Colab is really generous, so it's fine here now. And then we have training function, validation function, and testing function. Training function pretty much iterates over the small batches of the training data sets, and then performs forward pass, which is basically feed it into the model, and then computes loss, and then update the parameter. And the validation function pretty much do the same without updating the parameter. So it computes the validation loss without updates. And we're trying to select the ones that that has the best, which means the lowest validation loss. And then testing function is pretty much the same. So this is the technical way of doing it, a really common structure of doing it. Validate, pretty much the same. Test. Yeah, so just run it. And then it doesn't show anything, right? Does it show anything? Okay, it doesn't really show anything. Yeah, because it's just definitions. And then we are ready to train the model finally. So this 10 epochs means that we are gonna run over the model for 10 times. That means that the model is gonna see each sample or segments for 10 times. And then we are picking the model with the best one, which means that the loss is going to be the lowest. So you can press run, and you can kind of see it happening. So first it loads, and then for the first repeat for the first epoch, you see the training loss is a bit high, and the validation loss is like okay. And then for the second iteration, you see that the training loss has gotten lower. The validation loss is a bit lower, but not really much. And it's getting better. The validation loss sometimes pumps up, but that's okay. So sometimes happens. Yeah. So just a side note that I'm kind of lazy on the definition of validation loss. So you might want to use some alternative validation metrics. For example, like if you want to pick the model with the best, for example, like accuracy or F1 score, then it might be a better idea to try to use the F1 score or accuracy for your validation metric instead of using the same loss as the training loss. So you see that the validation loss has not gone down over the last few epochs. That means that we have a few overfitting going on here. So the training loss is eventually lower. And we have saved our best performance, which is after the second epoch. The validation loss is the lowest. And then you can see the effect of it by testing. So after testing, you can see the precision and recall of each category. So for AF, we have okay result, I guess, like 88, 80. For non-AF, we have ATA. Okay, it's pretty much the same. And we have accuracy and F1 to be about 85%. So that's like okay. So what you can do here, because we have 5 minutes left, and you can try to do some, mix and match on the transformers here. For example, like if I add random into it, and if I just remove these like standard practices for some reason, and then run all below, right? If you want to run all below, you can press run after here, or Control and F10, and then runs all below, and then you can see the performance, if the performance has gone up or not. And yeah, so now we have 4 minutes left. If you have some, maybe it's now for Q&A time. If you have some questions or things that you want to know about, you can be, asking now, I guess. Or we are gonna try to look at which combinations of transformations give the best results. Here we're running again, which does not seem better, honestly. And you can try to increase the number of epochs here to maybe like 20 or 60. It might have gone better than what we just did. And other side note is that if you change your runtime, if you did not change your runtime to GPU, like if you did stay on CPU runtime, then it's gonna take a while. It's gonna take like 20 minutes or something to run an epoch. So here it's taking like 11 seconds. So that's like, maybe not 20 minutes, maybe like 10 minutes. So it's a really huge performance difference. And hopefully this kind of give you an idea on how we usually do medical signals classifications on like using Python packages and using Google Colab. And if you wanted to use some more advanced techniques, then you probably would need, maybe you probably need more memory or disk space. So you can pay for Google Colab. So you can kind of see that if we remove the transformation, the align center and the baseline one removal and white noise removal, you can kind of see the performance has gone up, especially when with the randomness added into it. The randomness was added into it to prevent overfit. So that kind of shows as well, because since we have the lowest validation loss happening at epoch 9, which is a lot better in terms intuitively. Epoch 5 instead of epoch 2, right? So that means that the model is not prone to overfitting as much. So you can kind of adjust and try to mix and match and see what works for you the best. And this is like a short demo, and not all things are included into it. So just use it for reference. Okay, so this is on ECG data analysis. And yeah, thank you very much.